Can you hear me? Okay, awesome. Thanks for the introduction, Memming. So, I must preface this talk by saying that what we're about to discuss is by no means a new idea necessarily, and unlike a lot of the talks that we've seen up until this point that were very data-driven, this is going to be, for the most part, a fairly conceptual talk, so sit back and enjoy. This is more so in regards to a framework for a future line of research, that being a line of research that, despite all the pieces having fallen together, has not received the attention that I believe it deserves. So, with that being said, the title of this talk is Neural Dynamics as a Biomarker. Okay, cool. So, before we talk about neural dynamics, let's first talk about what is currently understood about biomarkers. So in 2016, the FDA and NIH coined the following definition for biomarkers, being a defined characteristic that is measured as an indicator of normal biological processes, pathogenic processes, or responses to an exposure or intervention. Now, somewhat of a very general definition, but that's sort of the point here. We can further classify biomarkers into various categories by use. For example, diagnostic biomarkers, as the name suggests, aim to predict if a patient is subject to a disease or affliction, and so on and so forth. So, biomarkers can stem from a variety of different modalities, many of which are molecular in nature. These are often categorized as either epigenetic, genomic, transcriptomic, proteomic, or metabolomic. So for neurological disorders specifically, we may choose to categorize them as neuronal, astroglial, or microglial. Often these are measured in the blood or CSF, just to name a few examples, for predicting the outcome of a traumatic brain injury or other acute neurological disorder. For instance, we may choose to look at, say, something like GFAP or S100 beta. Now, on the other hand, macroscale biomarkers also exist, and they take the form of, potentially electrophysiological recordings. For instance, we can look at the power band spectrum for diagnosing things like traumatic brain injury, or we can use imaging techniques to look at brain morphology, as, for example, dementia often exhibits forms of atrophy, as well as in many cases of schizophrenia. Now, I don't think I need to tell the audience here this today, but while keeping all this background of biomarkers in mind, the brain is notoriously complex, and this is due, of course, in part to things like network connectivity, tissue heterogeneity, as well as the fact that many neurological disorders are just temporally dynamic. So as a result, clinical neurological biomarker identification are essentially non-existent for many of the neurodevelopmental disorders, as well as many psychiatric disorders. And furthermore, there is certainly no shortage of individuals suffering from these sort of afflictions, with approximately 1 in 36 affected by autism spectrum disorder, 1 in 10 attention deficit hyperactivity disorder, and despite popular media portraying it as a fairly rare condition, 1 in 222 at least suffers from schizophrenia. And this slide isn't even including the common psychiatric disorders, with 2% of the population suffering from obsessive compulsive disorder, and unfortunately upwards of 5% from clinical depression, and that number is growing. So why is this happening? Why has so little progress been made in this certain specific area of clinical research? Well, first of all, neurodevelopmental disorders are incredibly heterogeneous in terms of genetic causes, rendering it extremely difficult, in some cases potentially even impossible, to identify a single biomarker from the modalities previously mentioned. In the case of psychiatric disorders, often treatment is evaluated and determined from behavioral screening weeks or even possibly months after treatment has been administered. This, of course, is a modality of biomarker that is subject to a lot of noise, as how an individual is even just feeling on the day of screening might totally throw off the results in this case. So this seemingly vast array of different problems that arise in neurological biomarker identification may, in some sense, to some seem insurmountable, but I want to make the argument that the problem is actually a lot more straightforward than that. Speaking simply, we are simply looking at the wrong level of abstraction. So neurodevelopmental disorders, psychiatric disorders, these are conditions that exist on the level of cognition, and thus deal primarily with high-level functioning. And what level of abstraction does high-level functioning exist in? Well, that would be the realm of neural computation, which is spoken in the language of dynamical systems theory, or neural dynamics. So now we get to what I believe is the fun part of the talk. So for those unfamiliar, don't worry, we're going to go over this in more detail in subsequent slides, and I'm also sure I'll repeat myself. So what's important to understand here is that neural systems that regulate a specific function often express computation by what is called metastable switching between attractor states. So let's break that down. What is an attractor state, or attractor for short? So an attractor state is a stable mode of operation for a system. So take, for example, a pendulum. If I take it, I let it go, and it swings back and forth for a period of time due to dampening effects, it will eventually come to rest at the bottom. That bottom state is the attractor for the pendulum system. Now, if I perturb the state of the system, being the position of the pendulum, it will eventually, in time, return back to this state. Now, the brain works in a very similar way. However, unlike the pendulum system, which only expresses a single attractor, the brain is highly nonlinear and thus brings about a multitude of different attractors simultaneously. Now, by metastable switching, what we mean is that due to perturbations of state, the neural activity will flow between these different attracting states in time, thus executing some desired computation. So these sort of perturbations of state can be caused by things including sensory input, thermal noise, as well as external effects such as things like microstimulation, deep brain stimulation, TMS, things of that sort. So one thing I'm going to be doing here, how do I get the pointer? There we go. I'm going to be referring to plots like this fairly often during the talk. These are phase portraits. For those unfamiliar, you can imagine that the state of the system is some point, let's say, in this space, and then we can imagine, say, a point in the top left quadrant. This represents some current type of neural activity in the system of interest, and then the autonomous dynamics, as indicated by the black arrows, will determine the evolution of that state in time. So if we start up in the top left, we will flow towards this black attractor here that I've indicated in the top left corner, and the state of the system will remain there unless perturbed by an outside force that can overcome the attractive force of that attractor, thus perturbing it over into the basin of attraction or attracting region associated with one of the adjacent attractors. So that being said, for those so inclined, you might want to think about this as a continuous time, continuous space analog of a Markov chain, as I've depicted here on the far right. So the main idea is that as this paradigm underlies pretty much, well, a vast majority of neurological function at some level, that disruptions to these neural dynamics fundamentally mean that these diseases are, at a very fundamental level, diseases of dynamics. So before I continue on, I just want to talk about the two types of perturbations that can occur in neural systems and more generally dynamical systems as a whole. So we've already talked about S-type perturbation. That's your perturbation of state, right? You have a neural system. It is expressed by some phase portrait and some global topology of different attractors. And you can imagine that you have a state, a point in that system that represents the current neural activity at some given time. And due to either things like sensory input or deep brain stimulation, you can imagine changing the location of that state within that phase space, thus changing the neural activity. On the other hand, you also have perturbations of dynamics or D-type perturbations. This is going to be a change in the actual structure of the phase space itself. So in this case, you could be creating attractors, destroying attractors, changing the topology of specific attractors. As I'm indicating here, the idea that maybe you have a point attractor, and by some means of D-type perturbation, typically as a result of learning or synaptic plasticity, you can imagine it turning into some sort of oscillatory attractor or limit cycle, where now the activity of the system is going to flow into this oscillatory state and thus continue to rotate around a region of phase space in time. So to really drive things home, I just want to use an example here. So I'm sure many of you are familiar with perceptual decision making. The modality of sensory input that we're dealing with for this doesn't really matter. I personally like to think about visual discrimination tasks, where you have an agent, they're looking at a screen, there's some collection of dots, and based on their average movement, either to the left or to the right, the agent has to make a binary decision as to which of the two possible directions the dots are moving, typically via a saccade. And this is somewhat of a toy example, but we can imagine some relevant set of populations of neurons, excitatory neurons, inhibitory neurons, that have, that are important for executing this task. And given an agent with these populations of neurons, by means of some learning rule, the agent has come to be able to perform the task quite well. And then we can imagine recording from this population of neurons, and we get some sort of neural recording as such, as I'm showing here in A. Now, I want to make it clear that the modality of recording, in this case I chose spikes, is not important for this argument. I'm simply using it as an example. It could be a variety of different modalities. This one just happens to be the one I want to use. That being said, one of the first things that we notice is that there is a lot of redundancy in this system, right? So during a leftward decision, we see that a whole slew of neurons light up. During a rightward decision on subsequent trials, we notice that a different slew of neurons all light up. And when there's redundancy in a system, that gives us room to simplify the problem, right? So in this case, we can imagine taking this 64-dimensional system, each dimension indicated by one of the neurons we're recording in this sort of paradigm, and instead talking about it as a neural trajectory that is transversing this low-dimensional state space or phase space. In this case here, blue trajectories are indicated with a leftward choice, red with a rightward choice, where gray trajectories are represent the resetting of that state in between trials. And we can go a step farther, right? So we can now talk about what are the underlying neural dynamics that actually generate and evolve this neural trajectory in time in this state space. And so just to blow this up a little bit, we can look at the plot here over on the left, and we see that the dynamics underlying this hypothetical visual perceptual decision-making task are as follows. So we have 3 potential attractors, one that encodes the state of the system in between trials, and 2 that encode both of the binary decisions. And of course here, as a means, like I said, of metastable switching as a result of sensory input from, in this case, the visual stimulus, we are perturbed depending on that stimulus into either the left or the rightward decision. But we could also imagine a case, right, where we have some D-type perturbation, perhaps from some failed bit of learning or some traumatic brain injury or some affliction to the brain that causes us to lose one of the attractors associated with one of those decisions, right? So in this case, an animal or agent or person that has this sort of system underlying this sort of perceptual decision-making task would no longer be able to properly encode one of the decisions and thus introduce dysfunction into the network, right? And this is ultimately what we mean when we say disorders of dynamics. The fact is that a lot of high-level cognitive functioning is the result of our ability to switch between these different sorts of attractor states, and thus by disrupting the global topological features, the attractors within that space, be it topologically destroying them, geometrically moving them in various places, or some other means, we can render that neural computation uncomputable by the system and thus introduce disease or dysfunction. So that was an example where we got rid of an attractor. Let's talk about an example where we create an attractor. So this is a toy model of the dynamics underlying one aspect of possible basal ganglion function here. So in this case, we have a set of attractors here on the right that are, under certain paradigms, believed to encode for possible behaviors that in a given context the agent is going to execute. And we can imagine introducing another attractor here on the left-hand side, rendering these attractors now marginally stable, or half stable, only attracting from one side. You can imagine in this case that the neural activity, if we start anywhere around here, will flow into its subsequent attractor, and then due to thermal noise or sensory input in the system, be perturbed out and into this oscillation here on the side. Now it just so happens that in this case, this is specifically a beta oscillation and thus serves as somewhat of a toy model for Parkinson's disease. I like this example because the beta oscillation that underlies Parkinson's disease, in many cases, is one attribute of electrophysiological activity that we've been able to notice with relatively crude methods. That being said, it's also of great evidence to this sort of idea of neural dynamics playing a part in a certain affliction. A lot of the paradigm that I'm discussing here today is really just saying that while this is a macroscopic example that, like I said, is already fairly well known, we also need to pay attention to a lot more subtler effects, a lot more minute detail within the neural dynamics that might bring about different afflictions. So we don't even have to go as far as, say, creating or destroying attractors in order to bring about certain dysfunction. So every attractor has their associated attractive force, right? How strongly they attract. They could be too shallow, they could be too deep. And under various hypotheses, some think that in the case of shallowness, now metastable switching is too easy for these states, right? Perturbative effects will cause the state of the system to escape that attracting regime in a much shorter timescale, causing it to jump unwantedly between other different states. And this is believed to be one potential idea underlying some of the positive symptoms for schizophrenia, where, for example, some have hypothesized that in primary visual cortex V1, this could lead to things like visual distortion. On the contrary, if you have attractors that are too attractive, they're too deep, now the metastable switching out of those states takes too long on average. And some have hypothesized that this may be an underlying or core cause of things like obsessive compulsive disorder, where depending on where in the neural system this sort of dysfunction appears, you'll see symptoms manifesting in different ways, with things like this occurring in the cingulate cortex, introducing difficulty switching between strategies, and lateral PFC, difficulty in shifting attention, and so on and so forth. Now, I want to make it clear, this, of course, is not the only hypothesis of neural dynamics underlying OCD. I guess this is somewhat of a redundant phrase. I mean, this is not the only means that people have thought that OCD might manifest, but the other hypotheses also have their roots in dysfunction of neural dynamics as well. So this brings me to kind of the core point of this talk, which is these three hypotheses that I want to put forth. The first being hypothesis 0, correspondence, that there are relevant differences between neural dynamics of healthy and diseased neural systems that capture the essence of the disease of neural dynamics, bringing us to number 1, characterization, that we can effectively characterize neural dynamics for healthy and diseased neural systems through closed-loop control. And this is going to be the topic of our next section of the talk. And that brings us to hypothesis 2, biomarker, that a quantifiable feature of the dynamics, such as shallowness, time constant of the attractors, how long it takes for that neural state to actually become sufficiently close to the attracting state, the number of metastable states that exist, topology of those states, how many there are, what is their geometric properties with respect to one another, with respect to themselves, state-dependent responses to stimulus. All of these are potential candidates that can serve as to robustly classify between diseased and healthy systems. So, I'm just gonna have some water. So one thing now that's important to talk about is, okay, so this is all well and good, but unless we can actually do this in practice, it doesn't really matter, right? So let me ask the question, can we do this offline? Can we take a neural system, record from it, and then do our analysis, get the underlying phase portrait, look at the dynamics, and then classify some biomarker from that? Well, the answer is no, right? Because neural systems, like I said, are incredibly nonlinear, and thus they bring rise to a multitude of different attractors simultaneously. Now, this in turn brings about biased sampling, right? If I have a neural system, I'm recording from it, and let's say there are 10 attractors in that system that are relevant for the specific computation of interest, and I've only sampled data from 2 of them because, let's say, for that animal or person or whatever, that they've only exhibited neural activity in a subset of those attractors. Well, then, via any inference method, be it online filtering or any kind of offline method, we're not going to have any indication that the attractors we haven't visited exist. And so that's depicted here, where we have this biorhythmic system here that exhibits 2 oscillatory attractors on the top and the bottom. And we can see that if we're using some online filtering method, that is inferring the dynamics via a recursive Bayesian method typically, that's using all the data from the beginning of the trial up until that current point in time, that there's no indication that the other attractor that we haven't visited is there. And so ultimately, this becomes an exploration problem, where we have to identify and sample from all the relevant attractors in the system. Okay, so the paradigm would have to look something like this, where we have some neural system, and then by means of several modalities of recording, in this case I'm just showing spikes and LFP, we take in that neural data, through some filtering algorithm, infer the neural state, it's a dual inference problem, the state of the neural system for that single trial, as well as the dynamics underlying the system. The dynamics, of course, being a better and better estimate over time as we get more data sampled from sufficiently many places in that phase space. And the real issue here, and the missing piece, is this maximization of exploration. How do we, at each iteration of this loop, stimulate the neural system to introduce an S-type perturbation in such a way that if we do this over and over and over again, we reach sufficiently many places in this phase space? And well, looking at the clock, due to time constraints, I'm going to run through this rather quickly. Can we do this with open loop methods? The answer is no, because open loop methods, the brain is state dependent. It's going to respond differently to stimulation, depending on where it is. Can we use classical closed loop methods? Again, we can't do this because we require some sort of a priori knowledge about the system, and that's what we're trying to figure out. I put model-based reinforcement learning, but really any sort of RL, and we can talk about this more later, but the general idea being that most reinforcement learning aims to maximize some sort of Markovian reward function. And you can show that the act of non-episodic exploration, non-episodic because we have to do this in a single trial, is intrinsically a non-Markovian process and doesn't fall into the framework of standard RL. And so we actually need something rather fancy. We need something nonlinear to account for the neural dynamics, also non-Gaussian too for that reason. Non-Markovian to deal with exploration, non-episodic because we have to do this in one shot, and non-stationary because we have this multitude of different attractors, right, that exist. And as a result of that, we need to account for all these different statistical anomalies in that, projected neural recording that we get from that low-dimensional latent dynamics. So that being said, this is our contribution for this problem. This is an algorithm that we've recently patented, Escape the Maze, paper to be coming soon. Let's talk about it real quick. The idea is that, generally speaking, we have an RL-like agent. That RL-like agent has an intrinsic reward function. We're using an intrinsic reward because in the case of exploration, there's no reward signal that's explicitly coming from the actual environment. Now, the idea of intrinsic rewards is certainly not a new concept. Plenty have existed before this, but ours is unique in the sense that, in a few ways. So first of all, I just want to point out that we went with something extremely simple and intuitive where the reward is intrinsically non-Markovian and therefore dynamic. As the agent explores, the reward landscape is going to change, and this reward landscape is going to be determined by, well, the reward that you get at any given time is going to be determined by where you are with respect to some distance metric that you can choose with respect to the closest point that you've already been to at any previous point in time. And so the idea is, let's say you've explored a whole set of spots in the phase space. Well, you can imagine exploring a new space up here, and that'll cause the reward landscape to diminish around that point. One might notice, too, that this sort of reward function may this reward function may increase in complexity as we continue to explore, both in computational complexity as well as memory, as every time we take a new step, we have to compare ourselves to every place that we've been prior. So we can overcome this by discretizing the space. We do this in the case of introducing landmarks. So in this case, every time I go to a new location sufficiently far from a previous landmark, I mark that spot, draw a landmark around it, and then every time you enter that landmark again, you increment a count associated with it. Time? Okay, I'll run through this super quick. Basically, more information about, theoretically, how Markovian rewards do not work in the context of exploration and that we need something non-Markovian. Let's look at some results. So this is a bistable system. The projection in 3D of a higher dimensional bistable system that we specifically made to model certain oscillatory behavior of traumatic brain injury, patients suffering from deep coma. And in this case, it was made to be very difficult, very challenging to transition between these two different states. And we see that current open loop methods only allow for local exploration, while our method allows for easy transition between the two possible states. If we keep going, we wanted to show that we could do this more than just in silico. So many years ago, back during my PhD, I was held down and forced to design this analog circuit testbed mimicking the same system. And then through a closed loop setup, as we indicated before, we wanted to show that this works in practice. So here's a video. Transition occurs when the purple line crosses the origin. Green here is stimulation. Yellow and blue are just, the other oscillatory states or dimensions in the system. So I'll let this play. Okay, and there we go. I cropped this fairly early, but the other methods that we tried cannot transition. Open loop methods, RL methods, closed loop methods, they don't work. You can run it for hours on this. I don't have time to show more results, but basically the gist being that every time that we throw this algorithm at anything, it typically explores several orders of magnitude faster than any other open loop or RL method that we tested against. And with that, I'd like to end my talk. Some acknowledgments. I'd like to thank everyone over at Reviver, everyone in the Computational and Theoretical Neural Information Processing Lab under Memming, as well as our collaborators, and also the funding sources that allowed this work to be possible. So with that, thank you very much, and I'm happy to take any questions.